Okay, so hi, my name is Erica. I'm a graduate student in the Gamillion Lab. I've been making videos on how to do cell culture protocols in case they don't exist in your lab or if you're brand new to the whole field. I'm making them to help teach my mentees and some students as well who are interested in cell culture. I will be putting forth what I have personally learned to do in the Gamillion Lab. And it may not be the correct answer. You may be told to do something else, and you should probably follow that if you have good justification on if that is still maintaining aseptic technique and if it's good for the cells. This is our biological safety cabinet here. And we have it in the same room that we do like benchtop work. And then we have like our student offices over here. And it's fine for research purposes. In an ideal world, you would want a biological safety cabinet to be maybe in its own room or even better in like a clean room. But we don't have that situation, and that's fine for what we need to do in our lab, right? Because of that, and because we're in like I would say, more risky space than usual, people in our lab need to have a more stricter and proper standard for aseptic technique, especially because we work with cell lines sometimes that don't allow for antibiotics or things that you add to the media to help protect against contamination. So I'm actually not making a typical media that we use in our lab today. I'm working with Rooster Bio's xeno-free media, which is 2 reagents. It is Rooster Basil and Rooster Booster, and I will be just adding these 2 together to create a xeno-free media. This, I would say, is atypical, one, because part of the protocol for Rooster Bio's media is to allow this to sit at room temperature for 4 hours, and then you combine it, and then you can heat it up in the water bath. I'm not really sure why, to be honest. I suspect maybe in their separate forms, it might be too much to put in the water bath. Maybe other people are actually putting it in the water bath, and it's fine. But I like to follow protocols to a T if I don't have reasons or justification for veering away from the steps described. So I have allowed this to thaw for a little bit now, but I'm going to continue thawing it. You are likely working with reagents that you can just heat up in the water bath. So you would just turn it on if it's not already on, place your reagents in there, and typically it takes 15 to 30 minutes to heat things up to 37 degrees Celsius, which you don't want to put anything colder than that really on your cells because you risk shocking them, and that will slow their growth rate and could kill them. If you're not planning on using the media immediately, you can just thaw it until it's liquid, and then that's sufficient enough to mix them together and then put it back in the fridge for later use. This is likely what the end product's going to look like. This is media that has phenol red in it, or an indicator that essentially tells you if your cells are becoming acidic. It'll turn yellow as the cells output more waste into the media to tell you as the researcher, hey, it's time to change my media. The xeno-free media will look clear as I'm going through this process, just to clarify that. It's because it's phenol red free, as to not disturb the HMSCs that I'm working with. So first things first, you want your actual first thing is to go pee. Do not be stuck in the hood when you have to pee really bad. It sucks. So you go use the bathroom, wash your hands, come back in here, and then put on your PPE. So for doing cell culture, you need shoes, closed-toed, long pants, a lab coat, safety goggles, and gloves. So I'm gonna go put those on. An additional 4th thing might be wearing a mask. If you're sick that day and you find yourself coughing and sneezing a lot, you might want to wear a mask at the hood any mask will do, just something to prevent you from sneezing or coughing into the biological safety cabinet, because that wouldn't be great. To just talk about aseptic technique when it comes to the water bath. I've talked about it in other videos that I've made, but I'll revisit it again if you need it. If you're heating up your reagents in here and you have a bottle in there, for example, and you want to now put it in the biological safety cabinet, what I like to do is to put down a paper towel on the counter, like so. Take out the bottle, let the water drip off, put the bottle here, so I'm not getting water on the counter. And then I dry off the bottle, starting with the cap and wiping away from the cap. If I wipe towards the cap, it risks sort of dragging potential contaminants up into the cap, especially if for some reason it's slightly loose. It shouldn't be, but this is all just safety measures to keep this not contaminated. And then you could spray with ethanol and bring it to the hood. Now I'm gonna set up the hood. You wanna open the sash to the indicator on the side. I open the sash to this line here so that it stops alarming and reaches laminar flow correctly. But you still have to wait for the biological safety cabinet to adjust to the open sash. Typically there's an indicator on the front that will tell you when it's okay to start working inside. Right now it's red, so I have to wait. But I can turn on the light. The light is green now. I also need to open my trash cans over here. You don't want to work with closed trash cans in which you have to touch them while you've got stuff in here that you're trying to keep not contaminated. So always open what you need. I always just by default open sharps and biohazardous waste, even though I may not be using the sharps. So I'm spraying my hands with large sprays. You should be spraying like that with everything to coat thoroughly. And now I'm going to spray my bench top with ethanol. Making sure to get all the spaces that I might be working with. I'm aware of where my arms are, sitting on the armrest. Always use the armrest so you're not blocking the airflow getting pulled in through the grate here. Okay, now I'll spray and add my things. Because I'm working with xeno-free media, I might need less things than you might need when you're following your protocol. It's important for you to read through the protocol and kind of like I do this. I'll make a working list of everything that I actually need to bring to the hood before I even come into lab, so that I'm ready to just fill the hood with what I need and be fast. I actually don't even need a pipette aid or serological pipettes or anything of that nature, because how the Rooster Bio media is that I just mix it together in the larger bottle, and then I can filter it if I want to, which I do, with a filter unit into a media bottle, and I'll show you how that's done. Again, fully spraying things, adding them to the back where the flow is best in the hood. And I'm making 510 milliliters total of xeno-free media today. A 500 milliliter filter unit will be able to hold this, so I'm gonna go ahead and get that out too. Spraying the whole bag. Place it in the hood. If your teaching lab does not have an in-house or inside of the BSC vacuum, you won't be able to easily filter using that kind of filter unit. In situations like that, it's honestly okay to just mix the Rooster Bio components into that bottle there. Seal it back up and put it in the fridge, or use it if you need to use it in the moment, because those things should be sterile. I'm just taking the extra step precaution and filtering with a 0.22 micron filter, and you need a vacuum to pull that liquid through the filter. So I'm going to open this filter unit by tearing this side that says tear here. Again, never reaching into a bag, always bringing it out to you so that your arms aren't touching the inside of the bag. In my lab, we put non-hazardous trash behind us and then pick it up after we're done using the hood, just to hurry and get out of the way. We don't keep a trash can by the hood just because the trash can will get full so quickly, and it's just easier to collect all the trash on the floor behind us and then pick it all up and put it in the normal trash can, pushing it down later after we're done. So this is our vacuum. I attach this to the filter unit, but I do not turn it on yet, so I'm just setting it up. And I also label what I'm about to put in this media with an ethanol resistant marker and some lab tape. I place the tape over one side that doesn't have the handles for like the indentations for your fingers to hold the bottle on it, just so the tape's not getting in the way of that. And I'm using an ethanol resistant marker so that when we spray it with ethanol in the future, it doesn't wipe off. I'm putting my initials, today's date. I'm putting what's about to go inside this bottle, which is xeno-free media. And I describe the contents of the bottle. So I'm gonna write that there's Rooster Basil and that there's Rooster Booster. Rooster Basil is 500 milliliters. The Rooster Booster is 10 milliliters. I write that too. The cap to the bottle I leave in the bag until I'm ready to use it. You could take it out at this point if you wanted and just keep it at the back of the biological safety cabinet, but I keep it in the bag just because why not? That's technically gonna keep it more sterile than if I open it now and don't use it immediately. If you have reagents in here that need to be pipetted into the top of the filter unit here, you want to do that for sure. Especially if you're not consuming a whole bottle, you don't know exactly what's inside the entire reagent bottle. It may say 150 on it, but it may have been manufactured with some error. So I'm gonna open both of these, place the cap with the closed side to the bench top. I know some people place a cap the other direction. I feel that the other direction is probably picking up germs and going inside of the cap, and then that is potentially going back into the bottle. So I place the cap side up like this. When I pick up lids too, face away from the palm. Don't turn it into the palm. And then now I'm going to add this to the bottle. This is a technique called sterile pour. So pour it in a way that you're not touching the opening of this bottle. This, you can put it to the side. And then I close this bottle completely. Mix it some. Not too much though. Bubbles will get created in this, and it's just not good to have that. And now I'm going to add this into the top of the filter unit. Again, this is not vacuuming yet. You want all your reagents in the filter unit before you start pulling. The reason why is, like, for example, with typical DMIM complete, which is probably what a lot of people are working with. If I add the DMIM first, vacuum that through, and then I add FBS, and now FBS is almost the only thing existing in this filter unit, the FBS is at risk to clog the filter, and then you're not gonna have a fun time. So you want everything to be mixed up here before passing through the filter so that you don't risk clogging the actual filter. Again, I'm doing a sterile pour. Cap this bottle, and I'm very careful not to knock this at all because it's very full. And I'm gonna open the vacuum on the side of my biological safety cabinet. I unscrew a knob over there labeled vacuum all the way. And now it's pulling through. And I'm just gonna add this lid back on top to keep it protected. So yeah, this yellow knob here is what I turned all the way open. Now it's pulling through. So if you're in a teaching lab situation, you didn't have a vacuum, you would be done at this point because you would have just put it in a bottle and then sterilely closed the bottle and then put it into the fridge. I still have my filter unit attached to this. So now I'm gonna disconnect the vacuum. I'm gonna close the vacuum. And now I'm gonna open my cap. I'll now remove this filter unit, and this whole thing can get thrown away. As I remove it, I'm closing this plastic lid all the way, turning it like this. Immediately you can put the cap on and screw it on this media. Now you have your media bottle. And this filter unit gets bleached and thrown away. I squirt bleach in this filter part and inside the vacuum pole itself, and then throw it away. These things can also get bleached as well and thrown away. Again, I'm not screwing on this all the way when I throw it away so that it doesn't pressurize in the autoclave. And now I've got my media bottle. Now, another thing I thought of, if any bleach happens to get on this stainless steel, you should definitely deactivate it with some ethanol and then wipe it up. Bleach will corrode stainless steel, and your PI or boss won't be happy if you have a rusted up biological safety cabinet. Another quick thing, this is not really ideal sleeves for culturing right now. Currently my other lab coat's getting washed, but I've been culturing for long enough now where I'm very aware of my sleeves, and I'm not hitting them on anything, or really the biggest thing is that I'm making sure that these sleeves are not getting caught on the armrest and knocking the armrest out of where it's sitting here. And that's just something to think about too, if that's a situation that you might be experiencing. It takes some time to build awareness for clothing like that, but just keep in mind that you don't want to be doing that or hitting your sleeve on top of an open bottle or anything like that. Yeah, now I can put this in the fridge and use it for later. This gets ethanoled and wiped down again. And with the vacuum closed, I can close the sash. And if nobody's using the biological safety cabinet after you, you're gonna want to UV light that. Final thing, important thing, is to also turn off the water bath if no one else is using it. You want to close the garbage cans to be compliant. You never want to leave biohazardous waste exposed like that. And I'm going to pick up some of the trash off the floor and throw it in the normal garbage. And that's it.